Okay, let's get started. Um, my great pleasure to have Professor Ed Dudley today to give us a seminar. And Ed and I were friends for many years,
Mhm.
and he's from Penn State University, food science. Ed has been doing a lot of research on Salmonella, E. coli, genomics work. So I would like to have him to take the stage to give us a seminar on his recent work.
All right. Well, thanks for that introduction, Wei, and thanks again for everyone for hosting me today. It's a pleasure to come out and talk about one of the projects that's going on in my lab. So we're going to do, if I remember this correctly.
You got it.
There we go. Okay, and you've got the full screen now?
We have a blank. Oh, there we go. There we go.
Okay.
You're good to go.
All right. So, okay. So, I'm going to tell you today about one of the ongoing projects in my lab that, revolves around this larger FDA project called GenomeTrakr, and some of the things that we've been doing as part of this consortium. But before I start, always like to give a shout out as well to the other members of the lab whose work I will not be talking about. And just pointing out also some of the other projects that we have going on in the the laboratory. So a big part of the laboratory also works on mechanisms of virulence of E. coli 015787, shown up here at the top right. Can you see the arrow? Do you have a pointer here?
I do not see.
Okay, yeah, that's fine. I just didn't know if there was a way to. All right, that should work.
There we go.
Yeah. So, a portion of the lab, works on basically what are some of the mechanisms that are driving production of Shiga toxin by 015787, particularly when it interacts with other members of the gut microbiota. Project mostly led by Dr. Aaron Rocky in the picture. And additionally, one of my other appointments here, besides the Department of Food Sciences, directing a 50-year center here at Penn State called the E. coli Reference Center that has about, to date, about 88,000 E. coli deposits. And one of the things we've been doing in the past couple of years is sequencing as many of these isolates as we can. We just broke 6,000 isolates last month. And we have some projects that are going on that are basically what are some of the things that we can do, given we have a large number of isolates, which we have both the genome sequence and the live bacteria. And one of the projects going on by Sharon Nieves Miranda, PhD student in the lab, is trying to identify some new serogroups or new O groups of E. coli using genomics. But what I'm going to talk to you about today is a project that involves not just us, but a larger consortia of groups, as I'll mention again and again, during the course of this talk. Tell you a little bit about this program, GenomeTrakr, for anyone who's not familiar with it. How Pennsylvania is integrating into this, and really three stories that I'm going to tell you about today. Is how we've used this as a way of, first and foremost, training our state labs, particularly our Pennsylvania Department of Health, in helping them get up to speed with genomic capabilities and how they can use these from a standpoint of epidemiology. Talk a little bit more about a ongoing project in the lab of how we are using genomics and salmonella to try to understand a little bit more about population structure of salmonella within wild birds and what this could mean. And lastly, we've been using this program as a way of kind of training the next generation. And I'll give you a story about projects that we have going on with the University of Puerto Rico around the area of antimicrobial resistance in both dairy and retail meats. So just to get us all started here, I kind of like to start with this slide, but for much of this crowd, this part is not news. You know that foodborne outbreaks are big news, in that when we often hear about these outbreaks, this is a particular one, large salmonella outbreak from about 12 years ago, initially linked to tomatoes. That was actually incorrect. The culprit was actually contaminated jalapeno peppers, but use this as a way of kind of introducing, often that when we hear about foodborne outbreaks, they're in a kind of a lay press or other release like this, in which, we pretty much at this point know what the culprit is. We typically know what food was responsible, and often how it got transmitted to human populations. And what we're often missing is the background story of how did we get there. And kind of just as a reminder that there's three different aspects to foodborne outbreak investigations. Optimally, we want all three of these. Often we only have two or sometimes one, so this chair is not often as stable as we'd like it to be. So the first are what we call traditional epidemiology methods. These would include things such as food surveys, trying to understand what are the people who got ill, what do they have in common, much more frequently than people who have been healthy throughout the entire outbreak. We like to eventually, if we can link it to a food, to an environment, to an event, we like to go out and just use traditional microbiology techniques to try to isolate the organism that we believe is responsible. And lastly, these techniques that collectively are called molecular epidemiology techniques, in which we're basically trying to assess genetic relatedness of isolates from the patients, isolates from the foods, in order to make matches that allow us to confidently say the organism from this food, for example, is what are making these individuals sick. And at least prior to March of 2019, the most common method that was used by public health laboratories, PFGE, pulse field gel electrophoresis, shown in the picture here, in which we were kind of just saying, generating a barcode, if you will, for different bacteria, and by comparing them, it allows us to make some of these conclusions. But since the beginning of 2019, and this is kind of what I'll focus on most of today, these molecular epidemiology methods have transitioned over to using whole genome sequencing, which provides us with a lot more information, a lot more conclusive. We're able to make some, much more definitive conclusions about matches. And, as many of us know, what was part of what was driving this, is also the cost. And this is a little picture that we take off the website for the company Illumina, which is one of the companies that, is heavily involved in manufacturing DNA sequencing equipment. Just communicating, one of the drivers of this is the cost of this technology has dropped precipitously over the last 2 decades, from one point in time in which it cost over $1 billion to sequence a human genome, to today it's less than $1,000. And of course, we've seen similar reductions in not only the cost of sequencing bacterial genomes, but also the amount of throughput that we're able to achieve. That's really kind of revolutionized the way that we investigate foodborne outbreaks. Another graphic to communicate this, and this is an image that I stole from Martin Wiedemann at Cornell University, and I know from the bottom he stole it from Amanda Conrad at the Center for Disease Control. But I think it very nicely communicates in a single picture the impact that whole genome sequencing has made on food safety. Using this example from Listeria, so we have a timeline from 1983 to 2015 going left to right. The blue line is the incidence of listeriosis in the US population per million. Clearly we can see it's been dropping over time. But in yellow, the bars are indicating the number of outbreaks that we've been able to define and detect. And at least on the surface, it looks like we're starting to see greater concerns in the food system with a larger number of outbreaks that are occurring. But the numbers at the bottom are really communicating what is going on and the impact that whole genome sequencing is having. That as we've proceeded, over time, the number of cases that are necessary in order to be able to identify an outbreak and understand that there is one that's actually happening has dropped, pretty rapidly, from about 70 individuals who were involved in a typical outbreak back in the 1990s to as few as 3 to 4 today. And a lot of this is being driven by the specificity and the power of whole genome sequencing, that it allows us to make some of these links between food and individual when there are just a couple of data points to compare. So it really has revolutionized, again, how we investigate and view foodborne illnesses. So this trend, as I said, whole genome sequencing is not something that obviously just came up within a year or two. It's been a trend over time where we've had these improvements. And back in about 2008, the U.S. FDA recognized that this is where the science was leading us, and had formed a program that they today is called GenomeTrakr. That started with a couple of labs, and again, today is a nationwide effort, actually international as well, that's involving well over a dozen FDA laboratories, close to 3 dozen state laboratories, 6 academic institutions, including Penn State, private companies, hospitals, and international partners as well, in which we're kind of working together as a consortia to try to advance the use of whole genome sequencing for epidemiological purposes, but with GenomeTrakr specifically for food safety. Now, when Pennsylvania joined this network in 2016, and really the expectations of a state when you join the GenomeTrakr program is actually, when you first come on board, it's actually just very simple. I mean, all they want you to do is to try and help them populate their public databases of genome sequences for the most common foodborne pathogens. And here's a snapshot, that I took actually a couple of weeks ago, meaning this is already, probably these numbers are already out of date. The website's down here if you don't know where to find the pathogen detection website on NCBI. But basically, it's a consortia, or a collection of genome sequences for all of these bacteria, and there's actually about probably 2 dozen others that are now included. So really, the only expectation is that you sequence a couple hundred isolates a year and contribute to this this database. But when Pennsylvania was approached about joining this network, we kind of took a step back and said, this is kind of a cool opportunity, not only for the state, but if we can make this as a collaboration between our state public health agencies and our land-grant university, being Penn State, we think we can actually make this a lot more powerful of a tool for us. So we started this whole process really with 3 core concepts in mind. First is we wanted to make sure that we were training our Department of Health as well as other state agencies on whole genome sequencing technologies, helping these individuals who had never sequenced or analyzed a genome before, help them understand how do we do this and how do you make sense of the data. The second is, instead of just sequencing random isolates and depositing them to this growing database, we wanted to deliberately sequence targeted collections that had some research question that needed to be answered, that could be answered by whole genome sequencing. And third, we wanted to always keep in mind that we have an expectation that we are going to be training the next generation of undergraduates, graduate students, postdocs, because clearly whole genome sequencing is the need for people who have specialized training in this area is fairly large right now and is only growing. So we're going to start off with a first story about Salmonella enterica. Again, this slide is not news to probably a lot of people, or most people on this call, but I keep this in here because sometimes I'm giving this talk to people who are very not very familiar with food safety. But it's going to be about Salmonella enterica, which we know many of these serovars have a very broad host range. And it's somewhere either first or second, around with Campylobacter as the most common bacterial cause of foodborne illness in the U.S. We know that there's over 2,500 different serotypes, also sometimes called serovars of Salmonella, based on the antigenic reaction to surface markers. But only about 100 of these have been previously associated with human illness. with Salmonella enteritidis and Typhimurium being the two most common. And I've also listed a number of outbreaks that have already happened in 2021. Again, just pointing out this is a very common, foodborne illness, and that we find Salmonella in a variety of different types of foods. So the first study that we did with the Pennsylvania Department of Health, just call it a retrospective analysis of isolates. So we were able to take advantage of the fact that our Department of Health is part of a larger program called NARMS, or the National Antimicrobial Resistance Monitoring System, which is a FDA affiliated program. I think there's about 14 states that participate in this, and Pennsylvania, shaded in purple, is one of the states that runs a retail meat surveillance program. So what that means is they routinely will go out to grocery stores, they will purchase chicken, turkey, pork chops, ground beef, and other, and try to isolate various pathogens, from them. Salmonella and Campylobacter get the majority of their attention, but they are starting to expand other pathogens as well. And when they do isolate these, they do some basic characterization, such as antibiotic resistance, assessments, and also, at least historically, pulse field gel, electrophoresis. So the Pennsylvania Department of Health has been part of this program, I believe, for about 13 years now. So they have a collection of bacteria that tell a story somewhat like we have here at the bottom, where they'll have an isolate that they pulled from a retail chicken sample that has, by two different restriction enzymes, has identical pulse field gel electrophoresis patterns to a human isolate that they have in their historic databases. This is a particular instance in which both of the isolates were also resistant to the same antibiotics. So we started this whole GenomeTrakr program asking some very basic questions of are in these cases where pulse field gel electrophoresis was suggesting a link, did this hold up when we're starting to use this much more powerful method, whole genome sequencing? So we did, this is a published study by this very talented master's student, Andrea Eiler, who is now working for the National Analysis and Countermeasures Center in Fort Detrick, Maryland. So one of the collections that she grabbed a hold of from the Department of Health was a Salmonella typhimurium collection, in which there was a isolate from a pork chop from 2013 that were matched to a number of historic human isolates in the Department of Health collection. So we call these strains SC01, which is the food isolate, all the way down to SC28. They were all from Pennsylvania, with the exception of one that came from a patient in Ohio. We already talked about the sources. PFGE, we can use one of two different restriction enzymes for this analysis. That for the most part, at least with the primary restriction enzyme, all of these isolates were the same, most of them by the secondary as well. And also, interestingly, antimicrobial resistance patterns were identical with all of these isolates. So the Department of Health had already published on this collection previously. And of course, what they're assessing with all of this data here with the pulse field gel electrophoresis, they were suggesting that maybe this pork chop was responsible for this outbreak, or something that was linked to that production facility. So Andrea took all these isolates, performed whole genome sequencing. Here's everything that I already told you, and constructed what's called a maximum likelihood tree, which basically just think of it as a family tree, a phylogenetic tree that's, by these various branches are just giving you an indication of who's most genetically related to whom. So I know it's a kind of busy of a tree, but basically just what's most important. We have our pork chop isolate SC01 up here at the top, and we can see that it is branched away from the most closely related human isolates. And in terms of nucleotides, or what we call single nucleotide polymorphisms, or SNPs, it was 41 SNPs distant from the most closely related human isolate. Now, as I mentioned earlier on in that initial outbreak slide, outbreaks are based on three different things. We can't often choose just either traditional or molecular epidemiology and make conclusions about whether an isolate was responsible for an outbreak. But what Andrea was able to figure out by looking at historic outbreaks of salmonella that were solved by whole genome sequencing is that at most, the salmonella isolates from food and patient were 18 SNPs apart from one another. So the fact that these were 41 is at least leading us to believe that this isolate, probably, was not responsible for all of these illnesses that were viewed. This little graph over here is just showing the antibiotic resistance patterns that were predicted genomically. And again, kind of interesting in that, at least one of these isolates had, genes that were not previously predicted. So really, as I said, very simple, study in which we've done many of these, like these with the Pennsylvania Department of Health, not only involving salmonella, but also involving Shigella, just kind of educating them about the power that whole genome sequencing can provide, for them. So the second story is led by, Dr. Yezi Fu in my group. This is a story continuing with the salmonella theme. That is a kind of again would not have been possible without the input of a lot of different partners, including the U.S. Food and Drug Administration, the Pennsylvania Department of Health, the U.S. Geological Survey, National Wildlife Health Center in Madison, Wisconsin, and the University of Georgia, Athens. So this story, it actually started with the USDA and the USGS reaching out to us and saying they've got this really interesting, salmonella collection. About 375 isolates that have come from sick birds in 41 different states over, in basically a 41-year period. And they'd serotyped these before to know that there are 46 different, serovars. Although the majority of them, about 68%, were Salmonella typhimurium. Salmonella Heidelberg being a rather distant second. And we'll actually come back to this one in just a little bit. So the question that the FDA and the USGS had was, how commonly are these salmonella carrying antibiotic resistance genes? And obviously just kind of wondering what is the role of wild birds in terms of spreading antimicrobial resistance. So Yezi, in a very an impressive short period of time, sequenced all 375 of these, and then just used some very common online tools, ResFinder, resistance gene identifier, to identify the resistance genes in this collection. What we found very quickly was that these isolates, at least surprising to us, tend not to carry a number of antibiotic resistance genes. In fact, many of them don't carry anything that's identified as an antimicrobial resistance gene at all. So 95% of these isolates are predicted to be pan-susceptible. Focus on the pie chart on the bottom, on the left-hand side for now. So we're looking at, where that represents all 375 isolates, is representing the resistances that we have found. So I said about 19% of the isolates, there was nothing that was even identified as an AMR gene. About 3/4 of them, we only identified one that goes by the very nondescript name AAC6'1A. This is a gene that is found in a lot of salmonella and is actually known to be phenotypically silent. So it's present, but it doesn't actually, you don't actually get a phenotypic resistance from this. It was only a few strains that we even saw any bona fide antimicrobial resistance genes, although noteworthy, we do have one that had 10 of them. But again, it was, I think, a little bit surprising to us because we were thinking these are isolates that are coming from animals that have been out in the environment, kind of like we see with pigs and bovine and other. We were expecting to see a pretty high level of resistance, but again, we didn't. That's not what we saw. On the right-hand side is kind of a developing story of Yezi's looking at the prevalence of heavy metal resistance genes, but that's one that's a story for a later date. But for us, it seemed like this was not going to be a really interesting collection. But Yezi kind of delved into this a little bit more, and very much to his credit, he came up with some pretty interesting findings. The first is in those 10 Salmonella Heidelberg isolates that I had mentioned. They'd all come from herring gulls, and they were all isolated in the early 1980s. All 10 of these isolates carried an antimicrobial resistance gene. These ones right down here, called phosphomycin A7, that was previously identified in a broiler farm in Canada. And this is a 2017 publication, but it was actually, these isolates were from 2012, and this was the first indication, or the first discovery of this particular resistance gene. So Yezi's study was able to push back, they were able to show that even 30 years before this study, that this gene had already been circulating in Salmonella populations. But the real interesting thing that came out of this has to do with phylogenetics. Before we jump into the data, I'm going to have 2 slides that at least make sure we are working with a common vocabulary here. So when we're talking about phylogenetics or relatedness, with not only Salmonella but other organisms, a couple of terms we use. So sequence type, or what I'll abbreviate ST, is a basic classification scheme, in which we are, looking at the allelic variants in specific, concert genes that are found in all members of, in this case, the Salmonella enterica. So by sequencing these and by looking at the allelic variants, we can place isolates into a sequence type that's given a number designation. Secondly, and kind of related, this was a method that was developed in my lab in around 2011, 2012. By Dr. Nikki Shariat, who's now at the University of Georgia, called CRISPR typing, which we use the CRISPR loci in Salmonella that tend to be fairly hypervariable. We can sequence them, and based upon the order in the sequence of these colored boxes that are called spacers, again, this is another subtyping method that allows us to separate isolates from one another. So those are the 2 terms I'm going to use when we start talking about Salmonella relatedness. The other, we have to talk very quickly about bird taxonomy, which up until Yezi started going down this road was all brand new stuff to me. So birds come in many lineages, as do bacteria. The passerines are actually the most common, or the most common classifier. About 60% of all birds. These include most of the things we recognize: perching birds, songbirds, cardinals, finches, and sparrows. Larids are coastal and water birds that include the gulls and the terns. The term waterbird is a different subset, including the cormorants, pelicans, and herons. And lastly, raptors are referring to many birds of prey, including owls, bald eagles, and hawks. So Yezi, when he sequenced, and this is just a subset of 131, Salmonella isolates in which we had the full metadata for. When he sequenced them and put them into a phylogenetic tree, so again, similar to what Andrea did in her study, but here it's actually shown in a circular format to make this a little bit easier to communicate. What he found was very interesting is, so first of all, these Salmonella isolates broke into 3 distinct lineages, in which it was not a random distribution, but the Salmonella clustered based upon their bird source. So there was a lineage for the passerine birds, one for the larids, and one for the water birds. And these are color-coded in blue, in yellow, and in light blue. So that 4th bird classifier, the raptors, those are the isolates that are color-coded in red, and basically they didn't really seem to cluster as tightly as we saw with these other birds. So the first kind of story from this is, it seems that the Salmonella group genetically based upon their bird source. If we move outward a little bit on the circle, focusing in on the 2 colored concentric circles on the outside. So the one on the extreme outside, that's basically green for most of the way around. That is indicating the sequence type of these isolates, and most of these isolates fell into sequence type 19, which is a pretty common, well-known sequence type of Salmonella typhimurium. Except for the waterbird isolates were a completely distinct sequence type, being sequence type 99. The circle that's just inside That's where we, are color coded based upon the CRISPR sequence type. And again, we see that it's not a random distribution. The passerine salmonella tend to have CRISPR types that are just color coded here, blue and gray. The lariids, tend to be a little bit more dispersed, but more the purple and the reds. And the waterbird are the sequence type that are color coded by green. So really, the punchline of this being, that salmonella among wild birds do not appear to be randomly distributed, but there is basic population structure. Looking at this a little bit deeper, so saying, let's not look just at our isolates, but can we compare these to a much larger collection of salmonella? So Yezi pulled about 1,600 salmonella genomes out of the public databases that came from sources other than wild birds, from poultry, from pigs, from cows, humans and food. Again, this is a phylogenetic tree showing the relatedness of everything and indicating that salmonella from the lariid and the passerine lineages form a very tight cluster in global populations of salmonella. And that water birds also form a tight cluster that's very distinct from those that are formed by the other two bird types. So it seems like even among a larger collection, this structure is holding up. So then we reached out to Dr. Xingru Deng, hopefully well known among many of you, a graduate of IIT and Dr. Zhan's lab. Because we knew from his previous studies, and he's very interested in using techniques such as random forests and other machine learning tools to try to make predictions of the source of pathogens just from the genome sequence alone. And the table at the top is data that he had previously published on, that again, just looking at the genomes of salmonella, is able to accurately predict the source of a salmonella correctly, being pigs or poultry, from 88 to about 91% of the time. Isolates that came from cattle, maybe a little less, 64%. But in this earlier study, they were looking at wild birds, and it basically was a guessing game. So only 52% of the time were they correct from the genome sequence for isolates that truly came from wild birds. And we actually see, looking at the numbers that he was comparing, it really had to do with there weren't a lot of wild bird isolates that were in the databases when he first published this study. The bottom is adding in, more of our salmonella isolates, and basically he's got the prediction, accuracy of wild bird isolates up to 83% now. So again, I mean, between the machine learning, between the phylogenetic trees and others, and the CRISPR typing and sequence type, we're generating this story that there really is a structure of salmonella that are found within wild bird populations. And Yezi's looked a lot more directly into the genomes, asking if there are some genes that are kind of predictive or might actually explain this grouping. And what he's finding that's interesting is it doesn't appear to be between these different bird types that salmonella have acquired new genes that maybe are helping them colonize these different hosts, but it appears to actually be more driven by pseudogenes. So an example of what he seems to find more commonly, salmonella that come from the passerine birds almost always are lacking a gene called FIMC, a very well-described fimbrial gene in salmonella. And the water birds are often missing a gene SAFB and STHC, which again are from two very well-described fimbrial operons in these organisms. So it almost seems to be much of the possibly if there's if we can predict that there's a biological reason for these differences, almost might seem to be due more to the loss of genes than gain. So wrapping up this part, one of the really interesting things that came out of this study, as he was sequencing isolates and was uploading them back last spring, we got a phone call from the U.S. Center for Disease Control, basically saying, we want to know a little bit more about these isolates that you're sequencing. And it was because they were tracking an outbreak. It wasn't foodborne, but it was linked to salmonella. And when they performed the traditional epidemiology, the surveys, they were finding that a larger number of people who reported getting sick also owned bird feeders. So they were starting to go down this path with this investigation that, maybe there was some link between salmonella being carried by wild birds, ending up in the bird feeders, and then people were getting sick when they were cleaning them. So we were able to share some data with them, do some analyses, and basically, I think it really helped strengthen their case that birds were really part of this particular outbreak, because our isolates that we were sequencing were the most closely related non-human isolates to the ones that are in blue, which were the human clinical isolates that they were studying. So it was just a really, I think, a really nice application. And I think it also really shows the power of what this GenomeTrakr program and making these large number of genomes publicly available, the true impact that it can have on public health in terms of being able to, define these outbreaks in a much more refined manner. So that's the second story. The last one I'm going to move over to now. We're going to now talk about E. coli instead of salmonella, and bring up these groups of individuals that we've been privileged enough to train at Penn State over the last couple of summers. So myself and a couple other microbiologists here down at the bottom, Dr. Josephine Wee, Daryl Coburn, Yasna Kovac, and Jonathan Campbell, have been funded for the last couple of years to run a undergraduate REU summer program focused on food microbiology. So we've had this so far in 2018, 19, we skipped 2020 for the obvious reasons, and we did this again in 2021. And the focus of it has been specifically to engage students who come from both small colleges and universities in Pennsylvania that don't have access to the same research resources as we have at Penn State, but also friends at the University of Puerto Rico, Aguadilla. Putting this into a little bit of context, so this is a ongoing project in which we're looking at antimicrobial resistance in E. coli, but doing this in the context of undergraduate education. So one of the things that was brand new to me when we got got some legs on this project, I had absolutely no idea that dairy is such a huge part of the economy of Puerto Rico. In this small island, there's over 200 dairy farms and over 40,000 dairy animals. Dairy is 70% of the agricultural output of the island. And despite the fact that we have something called the Veterinarian Feed Directive, which is a FDA regulation that basically prohibits the use of antibiotics in animals, except when it's under the purview of a veterinarian, antibiotics are still widely available across the island for farmers to treat their animals as they see fit. So that's kind of setting up the issue with Puerto Rico. Why the University of Puerto Rico Aguadilla, which is actually a small branch campus up here in the northwest part of the island, has about 3,000 students, but what really benefits us is over 30% of their student population are biology or other majors in the Department of Natural Sciences, so kind of fits very well with the goals. And also, the majority of the students at this campus are first-generation college and eligible for Pell Grants, so indicating a financial need and kind of the financial situation that many of these students are in. So we thought this was a great place to do some good science while helping out some of these students. So one of the studies that we've done has been involved a farm survey, 3 different farms in the northwest part of the island up by Aguadilla. One is a farm that raises beef cattle, in which the animals are fed graze, and the calves are fed by direct nursing from mother's milk. A second farm we just call Conventional Dairy Farm 1. Again, the adult animals are fed graze and grain. The calves are fed waste milk, which is basically the milk that comes from cows that are currently being treated with antibiotics. And because of it, antibiotic residues can end up in milk. The milk cannot be sold for human consumption, but can be fed to the animals for nutrition. A second conventional dairy farm we call Conventional Dairy Farm 2. Again, the adult animals are given graze and grain. The calves are fed from milk that comes directly out of the bulk milk tanks that's destined for human consumption. So in this one particular study, the students, over the course of a couple of months, they isolated 84 E. coli from fecal samples of these animals. They did traditional phenotypic antibiotic susceptibility testing, the Kirby-Bauer method. And then during the summertime, they come up to Penn State. They do whole genome sequencing on these, and we teach them how to do some basic analyses. So what was really striking out of this particular collection of E. coli is, so on the left is the prevalence of phenotypic resistance that we saw among cattle and the young calves among the 3 different farms. On the right is the resistance that was predicted from the genome sequence, so genotypic predicted resistance. And you can see really what pops out of this from this analysis is the calves from conventional dairy farm 1, so the ones that were fed waste milk, have E. coli that carry a much higher abundance of antibiotic resistance genes. Again, this was seen both phenotypically, and it was also predicted genotypically as well. So it kind of really pointing to the fact that this is an issue that's going on the island that we need to have been engaging some more dairy extension folks as a way of addressing. So the students who worked on this, Sharon and Paula. Sharon's currently a PhD student in my lab. Paula, we lost her to Ohio State University. They are the ones who did most of these analyses, and they also found that about a quarter of these E. coli isolates have one or more markers of the following E. coli pathotypes: enterotoxigenic, shiga-toxigenic, enterohemorrhagic, or enteropathogenic E. coli. They've learned how to do serotype prediction from genomics as well, and they found that the majority of the predicted virulent E. coli that they're finding were coming from the beef farm. A couple, fewer were coming from the conventional dairy farms. So that's kind of what I'm going to tell you about the research aspects of how we're leveraging GenomeTrakr to kind of advance genomics in a couple of different directions. And I hope I've at least convinced you. We're the 3 things that we are trying to do, both training our public health agencies, sequencing these collections that can tell some interesting research stories, and doing this as a way of training our next generation. And really where we're heading, in the future with this work, again, dividing these things up into 3. So the Salmonella, Pennsylvania Department of Health, study. So again, a lot of it driven by Dr. Nkucha Mikanafa, who's been a fantastic collaborator for the past 5 years. We're starting to move these studies to look more at salmonella and antimicrobial resistance in inpatient settings in Pennsylvania hospitals, looking at the patient outcomes given the salmonella genotypes that they were infected with and the antibiotics that these patients were treated with during the course of their treatment. Yezi's story on the U.S. salmonella isolates. He's now starting to actually branch out and reach out to international collaborators to try to put these isolates into perspective of other wild bird isolates from the United Kingdom, from Australia, and from New Zealand are some of the connections that he has right now. And lastly, I think one of the biggest wins from this overall project is Puerto Rico and some of the work we've done together. They were successfully able to get a grant to join the FDA NARMS program. So they are now going to be another site that's going to be doing surveillance of retail meats on the island, looking for antibiotic resistance. This is a grant in collaboration with the Department of Health and with Penn State as well. And this is going to supply much more kind of an even increased number of isolates that their students can sequence and analyze when they come up to visit us in the summertime. So that's pretty much what I have to tell you today about what we're doing with genome sequencing. And again, thank the entire group. Told you about Yezi's work. Naiduda, Zoe, Sharon, Aaron, and Allison, who all are on other projects, as well. But their work and energy is always appreciated. And in the work I told you about today, again, it's funded by the US FDA, the summer program by the USDA NIFA, and also the NIH is funding our E. coli work as well. So with that, I will end and be happy to take any questions. If I can just figure out how to close my screen down here.
You should have two tabs. One would show you presenting.
All right. Actually, escape work. There we go.
There you go.
All right.
Great presentation, Ed. I enjoy the wild bird part the most. It's so interesting.
And we've got 3 publications out right now on that one. One's been accepted in environmental microbiology, and that should be out soon. And then we have 2 that are currently under review. So hopefully that'll all be out in the public venue fairly shortly.
Yeah. Quick question. You didn't mention how they collect or isolate Salmonella out of the birds.
Yeah. So the U.S. Geological Society or Service, I always say society, it's actually the S of service. So this is one of their roles is to do both surveillance for various pathogens, but also they're one of the groups that when there is a maybe a sick bird that's been there's been some workup done on it, and salmonella has been isolated. They're often the group that ends up getting these isolates for characterization. So it's been because of their role in looking at avian health over the last couple of decades that they've been able to generate this collection. So yeah, I should actually qualify that for that second project. All of these isolates are coming from ill birds. So none of this has been just general surveillance.
So they actually kill the bird to get isolates from the organs?
These are, most of these are coming from either very ill or dead birds. Yeah, so presumably, I mean, it was the salmonella that caused this, but I don't, in all those cases, I don't know how certain they are about that.
Edward, kind of related to the same question that Wei asked, I saw that some of your students isolated their samples from stool. What were the main challenges for them?
Oh boy! So I mean, I think if you're talking about the E. coli, sampling, I think to them, maybe some of the main challenges were just getting their initial workflow together to be able to confidently isolate E. coli. I mean, it took them a little while to get a proper enrichment and plating method down, including where that project really started was that they were just interested in looking for S-Tex. And at least one of the commercial medias that they were using that claimed to be differential for various S-Tech serogroups ended up not being, so I think one of their biggest challenges was just figuring out from the tools they have available to them and some of these limitations, what's an interesting question that they can address.
Okay, Ping, do you have a question?
Yeah.
I think you're on mute, K-Ping. We cannot hear you.
Oh no. Is there a chat box?
Yeah, K-Ping, we cannot hear you if you want to type your question.
Okay, while Kaiping is typing her question in the chat, I have a general question out of the research part.
Yeah.
So at IIT, we're developing the undergraduate programs in food science.
Okay.
Can you give us a quick idea what's the size of the undergrad program right now at Penn State in food science?
Yeah, certainly. So right now we are at 160 undergraduates. In the 15 years I've been here, we've been as low as about 80, which is when I first arrived here. We got up to 330, I think, a couple of years ago, which, yes, Britt, that was a lot. So I think where we're at right now, we're probably starting to stabilize, a little bit in terms of the numbers that we're at, which, given the number of faculty we have and other things, is a pretty manageable number.
Wow, that's a huge undergrad program. I think it's way better than UW-Madison.
Yeah, UW-Madison, at least the last I knew, they're around 90 or 100. But they'll also tell you, if you talk to them directly, they deliberately are trying to keep it around that number because they don't they want, it's where they feel it's manageable given the number of people they have and some of the activities that they want to make sure they get students through.
Okay. Do you know at Penn State Food Science undergrad, out of this 160, majority of them from Pennsylvania or from other states?
Yeah, I mean, so Penn State in general. I mean, I don't remember the numbers off the top of my head, but the majority of students are coming from Pennsylvania. We do have a fair number who come from New Jersey and New York. Sometimes California seems to be another big draw. We certainly have been seeing an increased enrollment of international students as well over the last 3, 4, 5 years.
Interesting.
Yeah.
Kevin's question is typed in the chat box.
All right.
Yeah, I'm not sure if you can hear me right now.
Yeah.
Yeah, I changed the setting. Maybe it's the setting. Yeah, I'm just curious. And now, first, I want to follow up with Bazar's question. I like the 3 stories, especially the second one too. So when you tell us your student sequenced 375 isolates, how long does it take for him to do that?
That I made some comment during that talk of just how amazingly fast he did it. I mean, it was.
Yeah.
I don't remember, but it was no more than a few months. It was fast enough that we were supposed to be doing this work in parallel with the FDA. By the time the FDA got ready to ramp up and start doing this collection, they were like, "Wait a minute, you guys are already done, so it doesn't make sense for us to do any of this." I mean, when you think of it, with salmon, with everything that is done with the GenomeTrakr program is done using an aluminum iSeq, and with a genome the size of Salmonella, I know right now I think he's comfortably getting 35 isolates done at any given time. So I mean, it's not. As I said, and how you isolate the DNA, how you prepare a library, how you put it all on the machine. It's gotten to be so routine that it's really just more of putting the time and effort in rather than it being an overly burdensome type of technique. So kind of that whole thing of we can generate genome sequences now a heck of a lot faster than we can actually analyze them.
That's true. That's true. Yeah, I'm wondering, you collaborate with those state public health labs, and what is their workflow like? For example, they have their food items, contaminated food items, and they isolate the Salmonella, for example, from there, and then they prepare the DNA, and then prepare the library, run the sequencing, and then submit the sequencing result to GenomeTrakr, and they actually do their analysis on GenomeTrakr, or the FDA, the GenomeTrakr people do it for them.
Yeah, but so it's a little bit different. So with the Department of Health, and this is where, we kind of get into that, the FDA does it one way, the CDC does it another. And the CDC is, who's going to be kind of telling the departments of health of how to do things. So there still is if you're familiar with PulseNet, and basically this, tool in which, is used by all the public health labs for analyzing first, PFGE patterns and now whole genome sequencing, in which for the public health labs, they're basically, using this tool that integrates directly with the CDC to do all the analysis. The website I was showing you that, you can go out and publicly view all these genomes, that's part of the FDA's system,
Right.
Which is different from how the public health laboratories are doing their analyses. So, still the majority of the data that's being generated at the public health labs, just like before the whole genome sequencing era, it's not made publicly available. Yeah.
I see. Yeah. Okay. Thank you.
Yeah, you're welcome.
Any additional questions from the audience? How many mics do you have now
We just have one.
Oh, one.
Yep.
Okay, so if there's no more questions, I would like to thank you again, Ed, for excellent talk.
Thank you.
Nope, and thank you, for all the questions.
Yeah,
Okay.
this was wonderful. Thank you for joining us. This was great.
All right. Yep. Absolutely.
Bye.
Thanks.
Thank you so
All
much.
right.
All right.
Thank
Take
you,
care.
everyone. Thank you.
Thank you.
Bye.